What turns a cell into a brain cell, or a muscle cell, or a cancer cell for that matter? It is all about the DNA and what genetic markers get turned on and off. Scientists at the Salk Institute have created a 3D map of a cell that is giving them a unique view of how DNA works inside individual cells. Here's Kevin Enoch's report.
This new 3D imagery is letting scientists go on a tour inside the chromatin of a living cell. The chromatin is all of the genetic material, DNA, RNA, and proteins that tell each cell what to do.
The genome is the same in every cell in your body, but only certain genes are on in each cell. Okay, so obviously it's not the sequence, but it's the structure of these genes in each cell that determines if they can be accessed and used. So the question is then, what is the structure that determines if genes can be accessed and used? That really is what makes a brain cell, and a muscle cell, and also every human being different.
Using a specially created DNA dye, the team is able to see which DNA is active and which is resting in all kinds of cells. It is a completely new way of visualizing cells.
For a long time, people were able to basically study chromatin structure by electron microscopy. However, what people do is they have to basically take the cell and break apart the cell and look at the chromatin structure.
The idea is that seeing the cell in 3D can help researchers see how active and resting DNA work together in cells to make them unique.
And so what we're really trying to understand is the local structure and the global 3D organization, both in the nucleus and when it gets inherited across cell division in mitotic chromosomes. What are those structures? How do we inherit information? And if we understand those structures, can we now begin to actually manipulate it?
And by manipulating it, the researchers mean the potential to turn on and off particular sets of DNA, which opens the door to improved diagnosis and perhaps ultimately the ability to prevent diseases like cancer.